Students, welcome you all on the behalf of Rahul Anand Tutorials. So as you know that I am Rahul Anand and this is somewhat around my channel whose name is actually the Rahul Anand Tutorials. So now let's begin with the very interesting topics which are often being asked in terms of your need of in the Aims of in the All India Pre-Medical Examination. So these are literally belonging from which of the chapters of your book. So these are belonging from the the molecular basis of inheritance. So now let's start with the very interesting topics. So here we are going to study about what? The RNA, the ribonucleic acid. So when we are talking about the RNA, you have to understand the certain facts in it, such as what? So these are the single strand structure found here. Often when we are talking about these means the RNA, so these are not actually what? So these are not the genetic material, except in the some of the viruses, such as what? Such as the retroviruses. Fine. So when we are talking about these means the retroviruses are literally being found where? Can you tell me? So these are actually found where? So these are found in the here. In the case of the acquired immunodeficiency virus syndrome, as well as what? In that viruses. So that are probably known as what? So these are only known as the retroviruses. Now, often we talk about these means the structure of the here with the viruses in terms of the retroviruses. So these are having what things? So your retroviruses will be having the spikes like structure, playing an important role for what? Playing an important role to track down the helper T cell as well as the memory T cell. Literally directly proportional for the tracking of what? The the memory T cell. Otherwise, as well as by that mechanism, what will happen? Our immunity is literally being resolved as well as the level of the immunity are literally being descended. Why is it so? This is due to because, tell me, this is due to because it playing an important role to kill the helper T cell. Result in which what will happen? Our immunity will particularly become lower in level as well as it particularly descend. Often when we are talking about the structure of these the AIDS in terms of the acquired immunodeficiency virus syndrome, often the retroviruses. So these are having what things? So these are actually having the proteinous coat as well as the outermost coat will be having as well as known as what? So these are known as the P24. When you compare in terms of what? The inner surface of these means the acquired immunoviruses in terms of the directly proportional for the retroviruses. So this will be having what things? So this will be having the P18 means the protein actin coat in terms of its inner structure. Fine. Now, so these are particularly genetic material means what? In exception case of only the the retroviruses, as like when we compare the case of the DNA. So these are actually particularly all over the genetic material. Fine. Now, so when we are talking about these means the ribonucleic acid, you have to understand the certain facts in it, such as its monomers unit are known as what? Tell me. So these monomers unit are known as the ribonucleotide. Often when we are talking about these means the polymer unit. So this will be known as what? So this will be known as the polynucleotide in terms of what? The RNA. Often when we are talking about this, so it is often having the sugars. Fine. So these sugars are actually known as what? Of course the ribose sugar only. Now, so when we are talking about these, find here, the types of the ribonucleic acid or either the RNA, it will be popularly divided into the three broad categories. So these are actually what? So these are the, find here, the mRNA, then what? Then the tRNA, then what? Then the RNA, the ribosomal RNA, broad categories. So these are actually what? So these are the, find here, the mRNA, then what? Then the tRNA, then what? Then the RNA, the ribosomal RNA. So now let's understand about these, what are actually its role as well as introduce as well as the introduction in terms of these. Fine. So here, find here, the mRNA. So mRNA is known as what? The messenger RNA. As well as the provision about these means what are actually the mRNA, directly proportional for what? Directly proportional for the messenger RNA. So it actually provides the site for what? It actually provides the site for the protein synthesis. Often we are having the certain terms in it in terms of our messenger RNA. So these are actually what? So we will be having the transcription. Now let's understand about these means what are actually the transcription. Fine. So these are the whole wonderful process of what? Of formation of the messenger RNA in terms from what? In terms from our DNA in the deoxyribonucleic acid. So the process is literally known as what? So the process by which our DNA with the deoxyribonucleic acid will be converted into the mRNA. And it will be known as what? So this will be known as the here with the transcription. Often here we will be having the catalyst such as what? Such as we are having the RNA polymerase. Fine. In the second you are having what? So in the second we are having the another thing such as we have dealt about the mRNA. Then what? Then the tRNA in terms directly proportional for what? the transfer RNA. So these are actually what? So these are the carrier of amino acid. Find here. Thus, in this case, what will be happening during the protein synthesis? The tRNA will pick up certain of the amino acid as well as transfer it to what? As well as transfer it to the mRNA on the mRNA code on actually of the ribosome. So now, when we are talking about these, means what are actually the ribosome? So you are very clear about this. So what are actually the ribosome? Tell me. So these are the smallest cell organelles. So you are often clear about the cell organelles. So we will be having the certain parts in it, such as what? Such as the Golgi complex, the Golgi apparatus. Often we are having the certain parts, such as the ribosomes, the chromosomes, often the DNA, of course the nucleus. Often we will be having the certain parts in it, such as the Golgi apparatus, etc. Now, so let's understand about these, means what are actually the ribosomes. So ribosomes are what? So ribosomes are the smallest cell organelles. So the ribosomes are actually what? So these are the smallest cell organelles. So you are often very clear about what are actually the cell organelles. So we will be having the two different types of the cell in terms of what? The prokaryotic cell, the eukaryotic cell. Often we will be having the different types of the components in it, such as what? In case of the prokaryotic, the nucleus will be absent. In terms of the eukaryotic, we will be having the nucleus. Often you are having the certain types of the parts in terms of what? In terms of your cells. So these are actually what? So these are the, tell me, the nucleus. Often what? The ribosomes, the chromosomes. Often what are things? The endoplasmic reticulum, as well as the rough endoplasmic reticulum. Often the smooth endoplasmic reticulum. Fine. So these means are actually what? So these mean the ribosomes are actually what? So these are the smallest cell organelles. Often was discovered by whom? So it was discovered by the Padilla. Very important. Write in your copy. So Padilla was actually the scientist who demonstrated these, means the ribosomes. As well as these are oftenly being divided in terms of the two broad categories, such as what? Such as the here, means the 70S ribosomes as well as the 80S ribosomes. So the 70S ribosomes will be having the two subunits in terms of the larger subunit as well as the smaller subunit. The smaller one in terms of the 70S ribosomes will be what? So it will be having the 30S with reference when we are talking in terms of the larger subunit in what? In your 70S ribosomes. So it will be having the 40S. Fine. As well as literally when we descend in terms of the 80S ribosomes, mean the 70S ribosomes as well as in terms of the 80S ribosomes. So you will be having what? So you will be having here, mean the smaller subunit will be 40S, in terms of the larger subunit will be 50S. Often when we are talking the main major difference in terms of these, means the prokaryotic as well as on the eukaryotic basis. So your 70S ribosomes will be actually what? So your 70S ribosomes will be actually, tell me. So it will be belonging from the prokaryotic cell. As well as when we are comparing in case of the 80S ribosomes, so it will be belonging from what? So your 80S ribosomes will be belonging from what? Tell me, the eukaryotic one. So these are coming from the eukaryotic cell. So what are actually the prokaryotic cell? So the one in which the nucleus will be absent or either partially present, it will be coming under what? So it will be coming under the prokaryotic cell. With reference when we are talking about the eukaryotic cell. So these are actually what? So the one in which what will happen? So the one in which the nucleus will be present particularly as well as permanently, it will be coming under what? So it will be coming under the eukaryotic cell. So now, when we are talking about the amino acid as well as the proteins, so you are oftenly being clear about it. So what are actually the protein? Tell me. The proteins are the larger unit of amino acid. Then when we are talking about the amino acid, the reverse phenomena will be what? Applied. So amino acid will be what? So it is the monomer unit of what? Of actually our protein. So your protein are often being further divided into the four broad categories, such as the primary degree protein, the secondary degree protein, the tertiary degree protein, as well as the quaternary degree protein, often known as the 1 degree, 2nd degree, as well as the 3rd degree, then what? The 4th degree. Now, so primary degree, you are very clear about the rules. In terms of the 3rd degree, you will be having what? The quadrate protein, mean the fibrous protein, as well as what? As well as the globular protein. In terms of what? The tertiary degree. So in the 1st degree, we will be having the certain parts in it, as well as it represents what? So it represents actually the overall bending in terms of your DNA, as well as represent the sequence of the amino acid. With reference when we are comparing in terms of what? The polypeptide chain. In terms of the 3rd degree, you will be having the fibrous protein, the globular protein. What is fibrous protein? The one which are particularly attached with reference to point by point link. Often with reference, the best example will be what? The keratin present in terms of our hair, as well as what? As well as the ear, in terms of our nails. So these are belonging from what? The fibrous protein, in terms of the globular protein. You are having what? Tell me. So we will be having the pepsin, often the renin. So these are actually playing important role to neutralize the amount of the fat, as well as what? As well as break down the fats, as well as in terms of the coagulation. Means what? The protein as well as the lipase, important role. So you got that? Now. So let's proceed with the third one. Now let's understand about these, means what are actually the RNA, means the ribosomal RNA. So in this case what will be happening? So these are known as the most stable form of ribonucleic acid. As well as when we are talking about the formation, how these are actually being formed in the ribosomal RNA. So when your RNA plus with what? So when your RNA in the ribonucleic acid plus protein will be combined, it results in the formation of what? So it results in the formation of the ribosomal RNA. So you remember as well as understood about the proteins very well. So I have made you understand. So these are having the folding structure, often with the introduction in terms of what are actually the proteins. So I thought we have got what are actually these, means the ribonucleic acid, as well as these are not the genetic material. When we are comparing in terms of certain of the viruses, such as what? Such as the retroviruses, often in terms of the big introduction of what are actually the acquired immunodeficiency syndrome, as well as their introduction of the states, such as these are having the proteinous structure. Often you have understood what? Often we have understood about what? Often we have understood about the different types of the here in what are actually the parts of the RNA. So these are what? The messenger RNA, the transfer RNA, as well as what? The ribosomal RNA. So I thought you have got the concept. So if you like the work with reference to the education, often the lectures as well as the videos, so you could often subscribe, like, as well as comment below in the text. Thus I will be knowing that what concept actually you are hungry for, or what haven't you understood or either got. So very thank you. With reference to what? With reference to the Rahul Anand tutorial. So you could often write this. So very thank you.